I'm William Dowhan, Professor of Biochemistry and Molecular Biology at the University of Texas Health Sciences Center at Houston, the McGovern Medical School. My interests center on understanding how lipid-protein interactions influence membrane-associated protein structure and function. Although my early training was in protein chemistry and enzymology, I became interested in membrane proteins in the late 1960s due to a publication by Fred Fox and Eugene Kennedy that identified the membrane-associated M protein as the product of the LAC-Y gene of the lactose operon of E. coli. My postdoctoral training with Eugene Kennedy exposed me to phospholipid metabolism and enzymology, which led to my early work in purification and characterization of membrane-associated enzymes of phospholipid metabolism. My lab then focused on the molecular genetics of phospholipid biosynthetic enzymes in E. coli and Saccharomyces cerevisiae, which resulted in cloning of many of the responsible genes in these organisms. Although by the early 1990s many membrane proteins had been purified in an active form using detergents, there was little interest in the lipid environment in which these proteins reside. However, I asked the question, if lipid environment of membrane proteins is of little importance, why are there so many lipid species? To answer this question, my lab began to make null mutants in enzymes responsible for the synthesis of the major classes of phospholipids in yeast and E. coli. Although many of the mutants were viable under strict laboratory conditions, they displayed distinct phenotypes and have become a set of reagents to probe the functional role of lipids. After characterization of the in vivo phenotypes, we replicated the phenotypes in vitro using defined mixtures of specific lipids to establish a direct lipid-protein interaction responsible for the phenotypes displayed by the mutants. By altering membrane lipid composition, we provided evidence for the direct involvement of lipids in support of DNA replication, cell division, solute transport, energy production, cell and organelle morphology, topological organization of membrane proteins, and organization of higher-order protein complexes. Currently, my studies using E. coli center on understanding how membrane lipid environment determines the orientation of protein transmembrane domains. Studies in yeast center on the role of cardiolipin in organizing higher-order protein supercomplexes that make up the mitochondrial resperosome. There is still much to learn about the function of individual lipids in cellular processes. I am pleased that our work in demonstrating the importance of lipid-protein interactions has resulted in a shift of emphasis from studies of membrane proteins only in detergents to studies incorporating native lipid environments. Thank you very much for your interest and your attention.